So thank you so much for your interest in what Inscripta has to offer for your genome engineering workflow. So you heard our CEO, Kevin Ness, yesterday introduce our Onyx Digital Genome Engineering platform. So today, I will take you through the details of this platform and how you can use it in your genome engineering workflow and what applications the platform powers. So as biologists, our primary goal is aspiring to get to the best phenotype possible. And as Francis Arnold's quote says, evolution is a very, very powerful process in this. And we've come a long way starting from the slow process of natural selection that Darwin described, to getting to desirable phenotypes using targeted selection, such as breeding, for which Norman Borlaug got his Nobel Prize, to creating or engineering variation through directed evolution, for which Frances Arnold got her Nobel Prize. What has been our key learnings from both the targeted selection and directed evolution in how to get to that desirable phenotype? We need to generate a lot of variation. We need robust phenotyping of that variation, very rapid genotype to phenotype association, and finally combining them together to get to the next best phenotype, right? But to get to really rapidly improve the pace of biology or unlock its potential, we have to do this at genome scale. But engineering biology is hard. So we have a lot of strategies and tools already developed for protein engineering, but the question is, how do you go from precisely engineering proteins to engineering pathways and genomes to harness the power of biology? Because as you know, the space is really large. You can see the number of variants here, the permutations and combinations you need to go from protein to pathway to genome. So what do we really need? What we need is the ability to get to a large, broad variety of edit types, insertions, deletions, swaps across coding and non-coding regions, and the ability to deliver it across the genome. So what will that enable you to do? It will enable you to turn a bunch of biological knobs here, a diverse set of biological knobs, and you can test multiple hypotheses in parallel and at unprecedented scale. However, the current technologies that we have, they do a very good job of one or the other. So look at the axis here. The x-axis talks about the number of genomic locations. You have technologies and approaches available that will give you a single edit type like deletions across a number of locations in the genome. And you do have technologies, if you look at the y-axis, where you could select a single or a few specific targets and get to a variety of edit types. But to really scale this landscape and push genome discovery and genome engineering forward, we need new solutions. And these solutions really need to deliver on unprecedented scale, superior performance, and greater access. And that has been our mantra at Inscripta. So we've worked really hard, and we are really proud to bring to you our Onyx Digital Genome Engineering platform. And just when I want it to move, it will not. Thank you, guys. That was the introduction to the platform. So what is Onyx Genome Engineering platform? For all you practitioners out there, you know genome engineering is a very, very complex workflow. You need a lot of different things and competencies coming together to consistently give you good results. So the Onyx platform is a complete solution that has software, reagents, instruments, assays, and analytics optimized so that you can generate precisely engineered libraries of single cells at your benchtop at unprecedented scale, great performance, and extremely accessible to you. So what do I mean? I keep talking about scale, performance, access. So let's unpack that a little bit. What do we mean by unprecedented scale, and how do we achieve that? So the current technologies for scaling up genome engineering, what we see today is we use microtiter plates, right? So you have your guide RNA and homology arm, you co-localize them into 96-well plates. But at Inscripta, we wanted to take the scale much higher. So instead of using well-based partitions, we moved to cells as the partitions. So what that does for you is that you can now go from 96 to hundreds of thousands of cells that you can edit. But how do you get your guide RNA and homology arm into cells so that they are co-localized? We have a very simple and elegant solution. We covalently link the guide RNA and homology arm. And more, we also have a barcode, so now you get to a point where you have thousands of custom designs and millions of precisely engineered single cells, all trackable, that will give you thousands and thousands of phenotypes, right? So this is how we get to scale. But how do we make this accessible to you? So what I'm going to do is quickly walk through the workflow of how we deliver the genome engineering platform, how you can interact with it, and then I'll go into details of what is actually powering this platform. And finally, I will end with data we have generated on the platform to show you what applications and how you can use this for your own data, right? So the first step to any genome engineering workflow is choosing your hypothesis. What do you want to edit? So what are the targets you want to edit? So our platform has been optimized for E. coli and Saccharomyces. So you start with the edit targets. It can be specific or it can be genome-wide. And choose your strain or the Inscripta strain. And we start with the design part of the DGTL cycle. And I'm going to show you how we enable you to walk through the entire DGTL cycle and move through it rapidly over multiple cycles. It starts with the software. You come to our portal, you enter your targets, and what our software does is it will help you create designs at the push of a button. And you order reagents from our portal. The next step is the reagents reaches your benchtop in about 3 weeks. You load it on the instrument and go. Yeah, that's it. You just load and go. That's it. And at the end of it, you get a tube of edited cell library that is precisely edited and trackable. So many of you who visited our booth yesterday asked, okay, what do I do once I have the cell library, right? You have 2 paths. First, you can decide to see, okay, what is the diversity present within the library? How do I determine what the library metrics are, the performances? Or you can go directly to phenotyping. And once you phenotype, you need the ability to link your genotype and phenotype together. We enable both those steps because we've developed a portfolio of assays, which are NGS-based, that will help you determine both the cell library metrics and post-phenotyping very rapidly associate genotype and phenotype. And I'll go into details of all these assays in the next few slides. So once you've determined you have phenotype, and we do not do the phenotyping because you're all the experts in phenotyping and we really want you to explore a lot of phenotypes here. The next part is the learn part. So once you've generated a lot of phenotypes and you now have the ability to look at a lot of different data and data types, we at Inscripta have the analytics pipeline that will help you connect multiple experiments, learn from the experiments, get to the next hypothesis to speed up the pace of discovery, and power through multiple cycles of DGTL to keep improving the phenotype responses that you're looking at, or generating more knowledge from all the information you've generated here so far. So let's look at the individual components in some detail, and then I will tell you what powers all this behind. The starting point is Inscripta Engineer, which is a software portal that helps you design your edits very rapidly. So like I said before, what you see here is a library of about 1,800 designs in E. coli. And you can see that this is a Circos plot of the genome with multiple edit types mapped onto it. We have the ability, and if you want to know details about it, please come to our booth. We have a nice demo that we can show you of how the software works. But I just wanted to emphasize one fact. So you see multiple shapes and colors here. The shapes here indicate all the edit types that you can do and how you can do it across the genome. And the colors indicate the design score, because there's a lot of design rules in how a design is successful. And this indicates the design score, which is high, low, medium, or undesignable. So how do we know the design rules? We have very robust algorithms that is powering these design rules. So we've trained these algorithms over 300,000 designs across E. coli and yeast. And we've tested more than 1,000 features. Some of these features you're familiar with, right? It's what the community already knows, like diverse PAMs. What kind of PAMs do we use for the MAD7 nuclease that's powering our platform? So we have done a lot of analysis around it. We also introduce a few immunizing edits so that there is no recutting, and this helps with the edit efficiency. We look at cut to edit site distance, or how the template homology should be, that is involved in the repair. We are continuously doing feature improvements because we want to deliver the best edits and with a great edit efficiency. We've also tested multiple different edit types, insertions, deletions, swap at different edit lengths. And finally, we have created over 2 million designs so far using our platform. So what powers the instrument and the chemistry? We have a lot of proprietary technology, both in the chemistry, in the reagents that you get, and both in the instrument where there is microfluidics and engineering innovations. So why is this important? For those of you who have done genome engineering workflows on the bench, you do realize that the growth responses you see in the cells are very nonlinear. So how do you control for this so you don't have a complete skew in just one design taking over, or just the wild type cells taking over? We had to go through a lot of optimization in both the chemistry and the engineering microfluidics areas, and that's what the instrument helps control so that you get great edit efficiency. So I keep talking about edit efficiency and performance. So what are we actually measuring here? What is the metric by which we measure performance of our library? So one of the points I really want to emphasize here is when we talk about edit, we mean edits that are completely matching your design. So it's a very strict definition. We are very conservative in saying what is an edit. We call it complete and intended edit. So when you get your population of cells, you will have a fraction that contains complete and intended edits, but you will have some wild types and some partial edits too. Some of those might contribute to your phenotype, but at this point, we use only complete and intended to report edit efficiency. So we are quite conservative in that sense. 3 important metrics are critical for anybody working with libraries, right? So first one is edit efficiency. What is the fraction of cells containing complete intended edits in the population? The second one is design coverage. So if you have 100 designs you have input into the system, what is a fraction that you get out after editing that is very well represented? And the third one is design observed. What is a fraction of designs at least observed once? So depending on your use cases, you would be interested in all of these metrics. So how do we measure these metrics, right? So this is an important aspect. We have a portfolio of assays that you can choose from depending on your use case for phenotyping or understanding library metrics. So if you look at this illustration here, you start with a pool of engineered cells. That's what comes out of the box here. And then you can either go into a workflow that still continues as a pool, like maybe you're running some selections, or a workflow where you're isolating your cells. Maybe you're screening for a particular metabolite. In both cases, we have assays in which you can choose from so that you understand what is the genotype that you are either enriching in a pool or isolated with certain hits that you have in your population. All of them are NGS based, and we also provide the software to do the analytics, and I'll come to that pretty quickly. So the first assay that I want to talk to you about is the barcode amplicon assay. I mentioned that we barcode every single cell because it's trackable. So we have a plasmid-based assay that you can quickly sequence the barcode and bring the data back to our portal that then links your genotype to phenotype or gives you the design coverage metrics. If you want to really understand what is in your genome, we have an assay for that too. And it's an enrichment-based NGS assay that you could do. So that tells you exactly what edits are present in the genome. And the third one is, for those of you working with isolates, you might want to do whole genome sequencing, and we support the data coming out of that as well in our system. So we have a list of assays that you can choose from. And we support you with reagents, analysis, and data storage. So once you finish your assay and you generate the sequence, you can come to our portal, the Inscripta resolver, that helps you rapidly analyze your genotype. You can combine experiments here, and you can do both custom analysis, and we also provide dashboards in terms of assessing your run QC, like did my run work or not, which is a Sanger-based assay, and then all the edit assessment like edit efficiency or design coverage, designs observed, etc. So we help you with all the analytics. So I've been talking a lot about edit efficiency and edit performance. So how does the platform really perform here? How does the Onyx platform perform? So what you see here is a graph of edit efficiency, which is defined here as fraction of cells with complete intended edits in the population. The x-axis shows you E. coli libraries and yeast libraries. And the numbers here are the number of designs in the library. What is plotted here is the fraction of complete intended edits and other others you will find, like either wild type or partial or some errors you might find in the population. So what this tells you is we have an average edit efficiency of about 30% for both yeast and E. coli across different libraries, and it ranges anywhere from 15 to 75% because it depends on the targets you choose. Some of them may have fitness effects, biological. So we don't control the biology, right? We can deliver the chemistry and the optimization, but we don't control the biology. Another interesting fact you see here, and that is what I was referring to, is the first 4 of these are all there are about 100 designs in that library, and you can see that depending on the targets you choose, you could have slightly different performances. So what are some examples of demonstrated edit types, and you'll see this later in the data. So you can deliver edits in non-coding regions such as promoters or terminator swaps. You can also deliver edits in the coding regions, like you could knock out or truncate proteins using a triple stop, or do saturation mutagenesis across the length of the protein, or even in targeted sites. So what can you expect overall from our platform is we serve right now, it's single edit per cell for E. coli and Saccharomyces. You can either use Inscripta standard strain or your custom strain for these 2 organisms. We have an edit length that can go up to 60 base pairs. And for a given run, you can go anywhere from 100 designs per library to 10,000 designs. And we have a lot more that we can talk about the platform, and I would really encourage you to come to the booth so we can share a lot more information with you and see how the software and the instrument, we can demo that for you. So what I want to next focus on is the applications that you can run on the platform. And we have a lot of applications that we are working on both internally and with our early access partners. For instance, we are working on antibiotic resistance with Jim Collins at the MIT. We are working on stress responses with Marcus Herkard at the DTU Center for Biosustainability. And what I've marked in red here, we both here today and in Richard's talk tomorrow, we will go into much more detail on the data. There are two things that I want to emphasize here. One, You could be using just the native genome of E. coli or Saccharomyces to edit. Or you might have a strain where you have integrated and heterologous pathway that you can edit. So you can either edit the heterologous pathway or you can edit the pathway and the rest of the genome. So it depends on what your end phenotype that you need to get to, and that's the platform will help you in all of those scenarios. So this is it's a great talk by Richard tomorrow, so definitely don't miss that. It's at 1:30 in breakout room 2. So let's look at what's possible with the platform. Currently, the way we think about target discovery and optimization is either through classical mutagenesis or traditional strain engineering protocols. Both of them have great advantages, but also have a few limitations. Classical mutagenesis, the targets are pretty random. We don't have control over what targets get modified and what kind of modifications we make. While traditional strain engineering gives you very specific targets, but it's constrained by the scale of what you can do. So that is why when it comes to Onyx platform, we have to think differently. We should not be restricting ourselves to specific targets. You could do specific targets if you have a clear specific hypothesis you want to test, but you could also do completely unbiased genome-wide approaches. It also gives you the ability to turn so many different biological controls like saturation mutagenesis, knockouts, transcription factor binding site insertion, terminator swaps, promoter swaps. You can do a wide variety of things here. And finally, as I said before, what is important is not just generating the diversity and phenotyping, but also linking the genotype and phenotype together. And we have very rapid discovery processes with the assays and the barcodes that I explained before. So let me go through a few examples here to help you see what we can do with the platform. And I will start with specific targets, and then I'll move to genome-wide later. So the first one is the Inscripta resistome. For the E. coli community, we know the resistome is a very valuable database where the data of genotype-phenotype relationship to resistance to multiple compounds has been collected for decades. So what we decided is, how long would it take for us to recapitulate some of this years' worth of data that people have put together? So we went after the resistome database and picked specific targets that have resistance to multiple compounds, like 70 compounds across conditions, across 19 categories. And we recapitulated about 20 years' worth of data in 1 month in a single library. So one of our early access partners, Professor Chris Voigt, had a project with the DARPA that he wanted to look at linalool tolerant strains for a DARPA pressure test. Linalool is a plant terpene that's used for its scent, and I heard it's used to make beer hoppy. So what he requested is, okay, you have the resistome library. Could you add a few more specific targets to it? So we included saturation mutagenesis of some of specific targets he wanted. He tested them across the linalool, multiple linalool concentration, and we got 2 hits. So what you see here is your wild type MG1655 growth without any linalool, and then increasing concentrations of linalool, and you can see that the edited cells are performing much better than the wild type. So this is an example of specific targets. How do you now open this up to both specific targets and genome-wide? So let me give you an example here. This is an example of pathway and genome engineering of E. coli. We focused on the lysine pathway. So what you see here is DAPA, very important enzyme in the lysine pathway. How do you go from a protein like this? And then we went and selected a bunch of enzymes in the lysine biosynthesis pathway. And finally, we said, OK, let's go across the genome. So we made multiple libraries, which were saturation mutagenesis, knockout, and genome-wide promoter insertion. So when we say genome-wide promoter insertion, we did not insert just one promoter. We took 5 different promoters that had different ranges of expression and made 5 different libraries. Why stop with one, right? You have all the parts. So we made a total of 200,000 edits. Usually when you hear someone say you made 200,000 edits, the first question I get asked is, oh, so am I going to spend next 2 years just screening? No, because there are multiple ways you can actually get to the advantage of this without having to do a whole lot of phenotyping. Two things I would talk about today. One is selection, where if you have a good selection method, you're going to be able to use a lot of this and very quickly get to what enriches in your selection. Second is screening, and in that, I want to show you in the later example where shallow sampling will actually get you very fast to a good hit, and then you move on to recombining them. So I want you to keep that in mind as we go through the presentation. So what did we do? We took specific targets. Like I said, we did multiple libraries here, and then in unbiased way, we had other libraries. We pooled all of those together and treated them with a toxic growth inhibitor, which is a lysine analog. And once you do the selections on these different concentration, you use the barcode amplicon assay to determine what genotypes have actually enriched in your population. You'll see a lot more of the data tomorrow in Richard's talk, but what I want to show you here is actual counts from the barcode. It's a plasmid amplicon reading, but that's mapped onto the genome here that quickly tells you what is the number of counts you see in the control condition. So what happens when you put it through stress? Let's look at that. What you see mapped here on the E. coli genome is the enrichment score of all the stress testing that we did and did the barcode reading. The black dots are the pathway amino acid saturation mutagenesis. So there was at least one hit in every enzyme of the pathway of lysine biosynthesis that enriched under toxin selection. We found some more with genome-wide knockout. And one of the things I would like to point out here is the lysP. It is a transporter of lysine, and I'll come back to why I talk about it now. And then you have 100 new targets that we identified that is outside of the lysine pathway. So let's go from selection to screening, and I want to start with yeast. We did both yeast and E. coli experiments, and we went after high-value amino acids like lysine and tyrosine. Similar approach. We started with specific targets and did unbiased approach. We built about 44 libraries with 330,000 designs across E. coli and yeast, and multiple different edit types. So it's the same, build a lot of different edit types across the genome. So yesterday, Kevin spoke to you about a thought experiment. And I want to bring that back here today to say that was no thought experiment. We actually have data behind it. And what I want to show you here is the yeast chromosome, the 16 different chromosomes of yeast. We picked targets from the tyrosine biosynthesis pathway that's mapped in these circles here. These are the different enzymes we picked. We did saturation mutagenesis, 12 libraries, 88,000 designs. Okay, this is for the specific targets. Then what happens when you go in an unbiased format? We fill the genome with edits of different types, terminator swap, transcription factor binding sites, knockouts, 43,000 designs, and 7 libraries. So the point I want to make here is, yes, this is a large number. How are we going to phenotype this? Remember, I told you about the shallow sampling. So what you see here is just 1,000 edits sampled per library. It's only 8 libraries we've looked at. 42,000 total designs are in these libraries, and 1,000 edits sampled in isolates. This is not a pooled screen. This is in isolates. And you can see the libraries on the x-axis and the fold increase over wild type of tyrosine on the y-axis, and the different libraries marked in different colors. We see a lot of primary hits across. In the primary screening, we see a lot of hits in this. So I'll give you 2 examples. One for the specific targets, ARO4, which is a very essential enzyme in the tyrosine pathway. So what you see here is the ARO4 enzyme and the domain architecture for that, the residues along that domain architecture, and in the y-axis is tyrosine titer that we've measured using Agilent rapid fire mass spec. So what is mapped are all the residues that gave higher titers compared to the wild type. And the ones in purple are the ones that are previously identified, which is awesome because this is a great pathway that people have studied a lot, and we were able to recapitulate that. So it validates our technology. And then the orange ones are potentially novel mutations. So typically when you get to novel mutations in an enzyme, we map it back to the structure. So we mapped it back to the AR04 structure. Some of these residues fell in tyrosine allosteric binding sites, and some of them were in the substrate binding sites. But interestingly, there were several that were all over the protein structure, and we don't know what they do. So you can, even with a single target here, you're getting to a hypothesis that you can build and completely new knowledge. So what happens when you go across the genome? Do we find anything? Yes. So here is a graph with ranked set of hits that you found, and again, it's the titer, tyrosine titer. The highest hit we found so far was a genome-wide knockout of MET6. We don't know what it does. I don't know how it's related to the tyrosine pathway. There are several hits here. You could map back to a KEGG pathway or any of the pathway analysis tools that might tell you, yes, it's based off of carbon flux or some other hypothesis, but some of them are completely novel. We don't know what they do. And you can see that it's genome-wide knockout and just the terminator swap. So how does this look when you put it all back together in the genome? So you remember the previous graph of where the edits were all mapped, and what you see in these spikes are the hits, and the size of the spike is related to the magnitude of the hit in tyrosine that you're seeing. Even with very shallow sampling, we've already identified 25 unique loci. So this is the tyrosine story in yeast, so let's see what the lysine story looks like. Very similar, so I won't dwell on it. You're already familiar with the approach we are taking and the 200,000 edits we created. So let's look at the lysine. Again, we use the rapid fire mass spec to look at the tyrosine titer, lysine titer. Sorry, I'm getting confused here. The x-axis shows you the different libraries and the fold change in the y-axis. You see a lot of primary hits. We were able to recapitulate known knowledge, like DAPA is a very important enzyme, like I said before, and we recapitulate mutations in DAPA. But we also found a lot of new hits, like 17 new targets outside of the lysine pathway. Some of them, interestingly, lie in the Y-ome of E. coli, like the Y genome they call it, where 35% of it is Y-ome and nobody knows what they do. Those genes do. So we were able to identify this. So if you remember where I started, I said the fourth part after diversity generation, phenotyping, genotype to phenotype association, we've done all that. The next step is combining all these edits to scale that fitness landscape, right? So here's our first round. This is fairly familiar to the protein engineers in the group, I'm sure. So in the first round, we identified all these single edits, and some of them are highlighted, like DAPA, the amino acid residue change, DAPA promoter insertion, and a Lys-P knockout. So we took this DAPA amino acid change, and we created another library over on top of that, and boom, 10,000-fold increase in lysine production. So now you have DAPA amino acid change, DAPA promoter both recombined, you're driving expression, and you're making protein changes in the same cell. So why stop with 2, right? Let's go one more. We picked up that strain, and we did some of these hits from here and recombined them. So we found DAPA, the previous hit, plus the Lys-P knockout gave us another 4,000-fold increase. So in total 3 rounds, you get to 14,000-fold increase in lysine production. This is not anything new in terms of an approach, but the key point I want to make here is in a span of just 4 months and a very, very small team working, and most of the team was actually focused on the screening piece, we were able to get 14,000-fold increase in lysine production. So that was very exciting to us. But what is even more exciting that we are working on is truly multiplex combinatorial editing. The first product we are releasing is single edit per cell, but soon after, we are working already on getting to combinatorial. We are looking at 3 or 4 edits per cell, all edit varieties in both E. coli and Saccharomyces. So in conclusion, what really matters, bringing it all back together for unlocking the power of biology, is the scale, the performance, and most importantly, making it very accessible to all of you to be able to use the platform. The size and variety of edits and the trackability are also key. So the only thing you have to bring to the platform is your imagination and let that soar, like what Kevin said yesterday. Thank you. And we have a breakout session tomorrow, as I said before, and our Inscripta booth is 214. Please stop by and ask us a lot of questions. We want to hear from you. The more we hear from you, the more we will understand how the problems you can tackle using our platform. So thank you very much for your kind attention, and we might have time for a few questions. Sure. Do I take a mic here? Do they have a question?
Just wondering if you guys are...
One second. I'm supposed to wear this. Sorry, I forgot.
Are you guys working on mammalian cells, and how long until you get there?
Question, are we working on mammalian cells and how long till we get there? Yes, we are working on mammalian cells, but we'll probably announce that later in terms of the time.
My name is Yan Liang. I'm from Lawrence Berkeley National Lab. A very nice talk. Thanks.
Thank you.
I'm just wondering, can you explain more specifically, like, do you, like for your efficiency, how do you compare the efficiency of generating a knockout and compared to generating just like promoter swap or homology dependent insertion? Like, is there a difference in this efficiency for your kit?
Right, so edit efficiency depends on a lot of different, are you guys able to hear me okay still? It feels weird to talk with your headphone on. So edit efficiency depends on a lot of different factors, like what you pointed out, like knockouts may be sometimes easier than doing recombination. So this is one of the factors we are really optimizing in our platform to be able to deliver consistent edit efficiencies. And the key is also, like I said before, we don't control biology, so a knockout may be very easy to do, but if you select a target that is an essential gene, you may not actually retrieve the edit that you require. So there is a lot of different factors that go into determining your edit efficiency, and we try to optimize all of that to be consistent in our results.
Is there any biosecurity screening associated with this?
I'll probably shift that answer to Jason. Can I put you on the spot, Jason, about biosecurity? Because you just were in a panel, so.
Yeah, no, thank you. So it's a great question. Biosecurity is a very, very important aspect of our platform. So we do have a biosecurity protocol. We do screen incoming edits as well as protein coding region and potential outcomes from the edits.
Thank you, Jason. I didn't mean to put you on the spot there, but I did, so. So if you have more questions, please do come to our booth. We have a great team here from Inscripta that we are ready to answer all your questions. So thank you once again for your attention. Thanks a lot.